Thanks, Mark. I feel like I'm at the end of a speaking marathon here this morning. So, mooneye are a mystery to me. They're a species that I was not familiar with at all prior to my work here in the St. Clair-Detroit River system. So I thought it might be helpful to begin this presentation with just some background information on their biology and their life history. Mooneye are one of two extant members of the family Hyodontidae. The other one is the goldeye. They're presumed to occur in large, clear lakes and rivers, spawning in spring and primarily consuming a diet of insects. They're unique in that they are one of only two tongue-toothed species in North America. Google tells me that the genus Hyodon loosely translates to toothed tongue, so it's one of their identifying characteristics. Two of the others are the large eye, from which it gets its name, with a half gold, half silver iris and a small upturned mouth. Also somewhat unique to freshwater fish is the fact that their eggs float. They typically become mature between 3 and 5 years of age, depending upon sex. And in North America, this species is entirely exclusive to North America, and they have a range that's generally discontinuous. Our latitude here today in Bay City is presumed to be at the northern end of their range. And their historical range is a bit of a mystery and hard to pin down, because if you go back to some of the old literature, it suggests that there is frequent misidentification of these species with other similar looking species like gizzard shad, alewives, and some of the ciscos. So the discontinuous distribution that we see here makes mooneyes highly susceptible to extinction. They're presumed to be very sensitive to human-induced disturbances such as agriculture, urbanization, industrialization. And the fact that these populations are discrete, it means that they sometimes cannot be replaced or repopulated by local neighboring populations. So preserving the species is something that's important for the biodiversity of the St. Clair-Detroit River system. In Michigan, they are listed as an endangered species, and they are also a focal species of the Wildlife Action Plan for the St. Clair-Detroit River system. In the Great Lakes Basin as a whole, mooneyes have been found in Lakes Michigan, Huron, St. Clair, and Erie, although historically they seem to be more scarce in Lakes Huron and Michigan. Within the last 4 decades, they've only been found in relatively low numbers in the St. Clair-Detroit River system, which for the purposes of this presentation includes Lake St. Clair, its connecting waters, as well as its tributaries, such as the Sydenham and Thames rivers, which are circled here in the white oval. Contemporary biological data on mooneyes are even more rare than their observations. While basic information like length, weight, and age are available for some data from the 1920s and 1930s, little information has been available in modern days. Several of our modern reports of mooneyes come from anglers. As mentioned before, they're regularly confused with other similar looking species, so they are frequently the subject of email or internet forum threads that are, subject line, I caught this fish, what is it? In an effort to prevent occurrences like the one we have here on the right, I'm going to hazard a guess that fish did not make it. We're trying to create more of an awareness amongst our anglers about this species. Accordingly, because it's endangered, we have removed it from our master angler program as well as our state record fish program, since it is illegal to harvest, attempt to harvest, or possess T&E species in Michigan unless it's authorized by the DNR. So the contemporary biological data for mooneyes in the St. Clair-Detroit River system, again, is rare simply because they're only incidentally captured in our agency surveys. There are no targeted surveys that we have for this species. I went through our historical catch records at the Lake St. Clair Fisheries Research Station and found that we had caught 8 mooneyes since 1979. 2 of those were during the past decade, and both of those fish are pictured here. Talking to Ontario, they had caught 7 mooneyes between 2 recent surveys in 2018 and 2022. Similarly, they've been regularly captured by the ministry during walleye tagging efforts in the Sydenham and Thames rivers over the past decade. So appear to be a little bit more common in Ontario waters than they are in Michigan, but again, they occur in relatively low numbers compared to other species. So given that mooneye encounters are rare, their ecology remains a mystery. The left is just a photo of a fact sheet that I found on the webpage of the Michigan Natural Features Inventory. And when that was written in 2004, it highlighted knowledge gaps on mooneye, particularly population size, habitat use, and movement. Those knowledge gaps still exist today, but thanks to technology such as acoustic telemetry, the GLADO system, and agency partnerships, we now have the tools that we need to begin addressing some of these. So we're fortunate in that Michigan and Ontario have a strong partnership and demonstrated history of working together in the St. Clair-Detroit River system to answer questions that are related to the fish community. This project's another example of that. Michigan provided acoustic transmitters, Ontario provided equipment and local knowledge, and we both provided staff to begin some mooneye research that leverages acoustic telemetry technology and that existing acoustic telemetry receiver grid that's in place in the St. Clair-Detroit River system. Simply, I'm not going to dive too far into our objectives, but our overall goals are to learn about the when, where, and how these fish move. The timing, spatial extent, movement rates, and identifying critical habitats with an eye towards learning where these fish are at different times of the year, perhaps being able to develop a directed sampling program towards them and filling in some of those knowledge gaps. Methods were pretty straightforward. We used boat electrofishing to target mooneye in the Thames River, Ontario in late April of 2023. As Ontario's data from that system clearly showed that was probably the best opportunity that we had to lay our hands on this species. In about a day of electrofishing, we were fortunate enough that we caught 13 mooneyes, 11 males and 2 females. Those were measured for total length and surgically implanted with Innovasi V7 acoustic transmitters, which have a battery life of about 6 months. We had no idea what to expect tagging these, but we learned that they did handle the surgeries, including electroanesthesia, very well. All 13 of those fish survived and were detected on the receiver grid later. Our surgeons found that they were a bit of a unique species as far as actually getting access to the abdominal cavity to get those tags in, because the tissue on the keel of those fish is very, very thick. We did explore a number of tributaries on the U.S. side, hoping that maybe we would find some mooneyes, but we found nothing. And the striking difference that we observed between the tributaries that we sampled on the Michigan side and the Thames River is that the stream flow of the Thames River was just substantially more than any of the tributaries that we have in Michigan. So I'm not sure if that's something that might be attractive to the species, as they are presumably in that system spawning. So again, the white oval here is the Thames River. The star is where we sampled and released those fish upstream of the Chatham-Kent area. Interestingly, if you remember, one of the presumed habitat requirements of mooneye is clear water, and the Thames River just does not meet that criteria. So that's interesting to me. That's one of the things that you run across in the literature, is that they prefer clear water, and clearly here in the Thames River, that's nothing that's present. One of the other things we did was we removed a scale from each mooneye that we captured to non-lethally estimate age. For reference here, and I forgot to put the citation that I got some of these data from, but the light-colored bars on the left are from a paper by Van Oosten, I believe it was in the early 1960s, that provided length at age for some mooneyes that were captured from Lake Erie in the late 1920s. The dark bars over on the right are ages from the fish that we captured during our sampling in 2023. And you can clearly see that our mooneye were relatively larger and older compared to those fish that were captured during previous survey work. I'm not going to use the term average length at age here because many of these individuals or many of these age categories had a sample size of 1, simply because they're so rare. All of those that you see with the asterisk are age groups where there's only 1 fish sampled. So, hopefully with a little more sampling, maybe we'll be able to fill this out a little bit more better and look at some mean lengths and variability in age. But again, these fish, and especially the older individuals, are very infrequently encountered. So fun mooneye fact, just an aside here, I didn't split that previous chart by sex because we only capture 2 females, but it is very easy to distinguish male and female mooneyes. Male mooneye have a very, very prominent lobe at the front of their anal fin, as you can see on the left-hand side, that male individual. Females do not. Their anal fins are very concave, and it's very, very noticeable when you have these fish in hand to be able to just quickly determine their sex. As you all know, presentations about studies that use acoustic telemetry and animations of fish movement go hand in hand. You can't have one without the other. So this is a quick animation. If you could cue this up, please. It's the movement of mooneye in the Thames River, which obviously is not to scale here. It's much larger, just so you can see these fish moving back and forth. But the mooneyes were moving up and down the Thames River until June when they entered the lake. And so then data from that time to present are incomplete because we don't have all of the receivers downloaded yet. So we're awaiting those to be uploaded to the GLaDOS database to be able to learn a little bit more about their movements into the summer. Whoops. Let's see if we can. But yeah, you can see they go quite a ways up the Thames River and then back down into the lake and seem to congregate at the mouth there through the duration of the data that we have anyway. Let's see if we can click to the next. All right. So Dr. Vandegoot tells me that you also cannot talk about acoustic telemetry without showing the obligate Vemco hand. So here, here it is. The work that we did in 2023, I think, was an exciting proof of concept. So now that we know that mooneyes can be successfully captured and successfully tagged, we're going to continue the work in 2024. We're going to be using a larger V9 tag because the fish that were captured were clearly of sufficient size to handle that additional tag burden. And another benefit will be obviously the longer battery life of the V9s. I think we can get about a year of battery life out of those, which will hopefully allow us to provide data throughout all 4 seasons and capture a complete spawning cycle. So we would be happy to come back and provide that information at another time when we have it in hand. I would like to close by acknowledging the U.S. Fish and Wildlife Service State Wildlife Grant Program for funding the purchase of the acoustic transmitters. Thanks are also due to Andy Cook in the Ontario Ministry of Natural Resources and Forestry for providing us this mooneye capture data from the Sydenham and Thames Rivers. In our agency, Michigan DNR, Denise Alowski and Kevin Worley were instrumental in helping us to get these funds for the work. And I'd also like to thank Karen Soper with the Ontario Ministry of Natural Resources and Forestry and Amy Boyko with DFO for their assistance in making sure that we had the necessary permits in place to do this work. So with that, I am happy to answer any questions if there's time, and turn it back to you, Mark.